Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I begin the topic, please like, share, and subscribe the channel. In the last session, we dealt with stages of metabolism. In today's session, we will start with metabolic patterns of organs. The metabolic pattern or metabolic profile of different organs is different depending on its function. Moreover, the organs are able to adapt to metabolic alterations in fed state and starvation. Calories are stored in the body as fat and glycogen. The approximate percentage of storage form of energy, that is total fuel reserve, present in the normal human body is fat 85%, glycogen 1%, and proteins 14%. Fat stores are mobilized actively only on prolonged fasting, even though adipose tissue fat is undergoing turnover on a daily basis. Caloric homeostasis is maintained regardless of whether a person is well fed, fasting, or in a state of starvation. Similarly, metabolic profile of various organs and tissues change to adapt to physiological and pathological state. This is because caloric homeostasis is maintained unless extreme conditions threaten. The reciprocal regulation of glycolysis and gluconeogenesis is the major deciding factor in the flux of metabolic intermediates through these pathways. Although brain represents only about 10% of adult body weight, it needs 10 to 20% cardiac output. About 750 ml of blood circulates through the brain per minute. Neurons can survive only a few minutes without blood supply. Occlusion of blood supply to brain causes unconsciousness within 10 seconds. There is no stored fuel in the brain. Glucose, the preferred fuel for the brain, should be in continuous supply. Glucose can freely enter the brain cells. The total consumption of glucose by brain is about 120 gram per day, that is 480 kilocalories. Thus, about 60% of total carbohydrate intake by the body is metabolized by the brain. Moreover, about 25% of the oxygen consumed by the adult body is due to glucose oxidation in brain. In children, this may be as high as 50%. Coming to brain under conditions of anoxia. In anoxia, the rate of lactate production is by glycolysis rises to 5 to 8 times per 1 minute. The Pasteur effect is the brain's protection against conditions of anoxia. Blood glucose level below 30 mg per deciliter is fatal. Coming to brain and acetoacetate. The brain is unable to utilize fatty acid as a source of fuel since the fatty acids complex to albumin are unable to traverse the blood-brain barrier. But brain can effectively utilize acetoacetate. This is again a survival technique in diabetic and starvation ketosis. Coming to brain and starvation. During starvation, a significant part, that is 60 to 70% of the energy requirement of the brain is then met by the ketone bodies. In this diagram, we can see liver and brain. Brain under fed condition uses glucose as energy, whereas in starvation it uses ketone bodies as energy. Under conditions of partial anoxia, the production of ammonia is increased. This is immediately trapped as glutamine. The amino group of glutamine and glutamate can be used for synthesis of other amino acids. Coming to skeletal muscle. The skeletal muscle forms about 45% of the total body weight. About 0.5% muscle weight is due to glycogen content. Following a meal, the muscle glycogen content increases about 4% of total weight. Coming to muscle metabolism after meal, the uptake and storage of glucose by the skeletal muscle is under the influence of insulin. Following a meal, the level of the glucose and insulin are high, so glycogen synthesis is enhanced. The resting muscle uses fatty acids as a major fuel, that is 85%. Coming to muscle metabolism during exercise, muscle uses glycogen for short active spurts of activity. In this diagram, we can see that in the fed state, muscle and brain both uses glucose as a source of energy. Glycogen is rapidly broken down to form lactate. The lactase has to be transported to liver to undergo gluconeogenesis. Muscle, however, uses fatty acid as fuel for aerobic exercise and long distance running. Coming to muscle metabolism during starvation. During starvation, maximum glucose is shared for brain. The free fatty acid, that is FFA, mobilized from adipose tissue is the preferred fuel for muscle during starvation. Free fatty acids does not require insulin, and during fasting, insulin level is also low. In this diagram, we can see that major organs are arranged horizontally, whereas stages of metabolism are arranged vertically. During prolonged starvation, muscle protein breakdown occurs and alanine is released to the bloodstream. It is transported to liver and provide substrate for gluconeogenesis. The metabolic fuel during prolonged fasting is ketone bodies. Branched chain keto amino acids are utilized by the skeletal muscle. Coming to the adipose tissue, it is the storehouse of energy in the body, rather 135,000 kilocalories. The energy is stored in the concentrated form, that is triacylglycerol. In chylomicrons and the chylomicrons and VLDL are hydrolyzed by the lipoprotein lipase present on capillary walls. It is activated by insulin. The fatty acids are re-esterified to form triacylglycerol. The glycerol is derived from dihydroxyacetone phosphate, that is DHAP, an intermediate of glycolysis. Therefore, for storage of triacylglycerol, both fatty acids synthesis and glycolysis should operate. For the uptake of glucose, glycolysis and lipogenesis are all favored by insulin. About 25% of glucose taken by adipose tissue is metabolized by the HMP shunt pathway and the rest by glycolysis. The NADPH generated from the shunt pathway is used for the synthesis of fatty acids, whereas NADH produced during glycolysis is used to reduce the DHAP to glycerol 3 phosphate. During fasting, triglycerides in the adipose tissue are hydrolyzed. The cyclic AMP mediated activate hormone sensitive lipase occurs in response to the high glucagon insulin ratio. Moreover, glucocorticoids have a stimulant lipolytic effect during fasting. Coming to liver, the liver plays a central role in metabolism by providing adequate quantities of metabolic fuel for other organs. Almost all the metabolic pathways operate in the liver, notable exception being ketolysis. Coming to liver metabolism in fed state. Under well fed conditions, the liver takes up glucose and from circulation and stores it as a glycogen. Similarly, the fatty acids synthesized by the liver are incorporated into VLDL and secreted into bloodstream. Liver is the major site of degradation of amino acids and detoxification of ammonia into urea. During starvation, liver produces glucose by glycogenolysis and by gluconeogenesis so that the obligatory requirement of the brain are met. In this diagram, we can see that free fatty acid is converted to ketone bodies which is taken up by brain for energy. Also, muscle protein breaks down and is released which forms glucose, and again brain takes up this glucose for energy. Moreover, liver also produces ketone bodies, an alternate source of fuel, but the liver cannot use ketone bodies as its own fuel. Coming to cardiac muscle. Heart consumes more energy than any other organ. It utilizes about 6 kgs of ATP per day, that is 20 to 30% of its own weight. Cardiac muscle derived is its energy by oxidative metabolism of fatty acids 60 to 90% and glucose which is 10 to 40%. Ketone bodies are also normally metabolized. In this diagram we can see that creatine kinase helps the generation of ATPase. This ATPase has three functions: sodium potassium ATPase in the kidney, myosin ATPase in the heart, and calcium ATPase in the muscle. In addition, energy transfer to heart's myofibrils occurs by creatine kinase catalyzed by energy shuttle. The phosphocreatine, in being a smaller molecule than ATP, and can easily diffuse into the myofibril from mitochondria. The myofibrillar creatine kinase catalyzes the reformation of ATP. The free creatine diffuses back. The creatine kinase system acts as an energy buffer by keeping ATP level constant. When ADP level increases due to a fall in phosphocreatine, it inhibits intracellular enzymes, causing failure of heart's contracting mechanism. In a failing heart, the uptake and utilization of fatty acids and glucose occurs, whereas in advanced heart failure, insulin resistance develops. This decreases the glucose utilization. At the same time, the metabolism of hypertrophied heart switches from fatty acids to glucose. Here ends our session. Kindly like, share and subscribe the channel. Thank you.